Okay, hi everybody and welcome to our lab today. So our lab this week is going to cover nervous tissues, a little bit of physiology, and then because they robbed a week from us, and then I was going through my stuff and realized that you would have a whole lot of stuff in that very last week if I put the lab practical in the last week of school as well. So I've moved everything up while I kind of am combining some stuff. So I'm going to do some combining with another week today. That's in the, but it's in the yellow packet. So you guys have all the information that you need. Let's see. What was I going to say? Why I wanted to get this recording before. Oh, I know. Make sure you get your quiz done today. It's due today, so to April 7th. So lower extremity muscle quiz. Get that taken if you watch this today. If you didn't watch it today and you didn't get it taken, bad. All right. So oh, and here's the other thing. We still are having the blood drive on Thursday. I don't know if I mentioned it to you guys because I know I mentioned it to 202. So if you are around and you're able to give blood, please donate blood. We're really in need of blood. And in fact, I will give you 20 extra credit points if you go and donate blood. So I will put an announcement in. You need an appointment, and I will put the phone number for the Red Cross so you can get an appointment in the announcements as soon as I load this in the recording into the announcements or into Canvas. And then make sure that you sign up on the sign-in sheet that is at the that will be at the blood drive. I'm not taking like, oh, you know, here's whatever. No, you need to sign on that sheet in order to get credit for donating blood. But I'll put that in the announcements too. So there that is, in case you're able to do that for us. All right, so we're gonna go to our yellow packet today, and we're gonna be in week 11 that says nervous tissue and physiology, and then the homework that goes with that will be exercise 15, 16, and PEX3. Excuse me. All right, so we're gonna start out with neuroglial cells. So let me find those, there we go. And so we're in our lab manual on page 12, 14, 15. Right, so we're on page 252 in the lab manual. So always, it's a really great idea for you to bring your lab manual to our meetings or while you're watching this, so that I'm just making room for mine out here. Oh, you guys, I just chuck things all over the place. All right, so we're gonna first look at neuroglial cells. So I'm gonna pull that up on my thing and then share that. Oops. And away we'll go. So, oh, let me, hang on. Let me make us big. All right, now we'll share. Okay, so let's get rid of that. Okay, here we go. So, oh, wait. I don't know what I'm sharing. Not what I wanted. There we go. Okay, so here we see the astrocytes. Okay, so this group of 5 cells right here are known as neuroglial cells or neuroglia. And what that means is nervous glue. So these are cells that we find in between and around with the neurons that are not going to carry nerve impulses. So make sure you know that neuroglial cells do not carry nerve impulses, and they do a lot of other supporting functions for the neurons because the neurons are so specified, so particular to their function that they can't do much of anything else. So we're going to discuss important things, and then we'll review this from our lecture notes tomorrow, and we'll do our Kahoot, and then we'll be ready for our test on Friday or by Friday. But I'll probably open the test up for Thursday and Friday of and at least for now, and then we'll see if I add any more days to that. Okay, so neuroglial cells. The very first one, I'm just going to scroll through these. The very first one is an astrocyte. So for the quiz, you need to be able to recognize that this is an astrocyte right here, but for the lecture exam, you need to know the function. So I'm going through the functions as well. So just learn it all, and then you'll know them by sight, and then you'll know them by function. Okay, so astrocytes, astro means stars, so they look like stars. They've got all these little, or a sun, of these little extensions that come off of them. And what these extensions are going to do is wrap around the axons and dendrites of a neuron, and then they're going to wrap around the capillaries. And so what that does is provide a barrier between what might exit the capillary and what might get into the interstitial fluid or cerebrospinal fluid that's surrounding the neuron. So the astrocytes form what's known as the blood-brain barrier, and it's in your lecture notes. And like I said, it's going to keep material out of the environment of the neurons so that they aren't harmed. But unfortunately, there are some things that can cross the blood-brain barrier. And also, when we are engineering a drug that's going to be used like at a synapse or something, that we want it to be able to cross the blood-brain barrier. Yeah, I mean the exam on Friday. Yeah, so the lecture exam is our regular, test number 4. Okay. All right, then another function, well, also let's look at this down here. Astrocytes are the most abundant central nervous system neuroglia. So we have more astrocytes than anything else. Also, astrocytes and the other neuroglia have the opportunity to replicate themselves, go through mitosis. And so when we have brain cancer, it will be a glioma. The tumors that we have in brain cancer will be caused from excessive growth of the glial cells, or so we call them gliomas. What was the other thing? When we need to get rid of the neurotransmitter that is in the synapse. Remember, we said we have to terminate those neurotransmitter effects. One of the means by which we can get rid of the neurotransmitters is that the astrocytes will take them up as well. So I put in the notes, it mops up neurotransmitters. And also when we hyperpolarize the cell and we have too much potassium on the outside of the cell, okay, because lots of potassium leaked out to make it hyperpolarized, we can mop up the potassium to get rid of the hyperkalemia and restore membrane potential again. Okay, so those are the astrocytes. Okay, the next ones in our picture, not on our list, but the next ones in the picture are the microglia. It says microglial cells are the defensive cells in the central nervous system. They are phagocytic. So these are cells that can go around and eat up other things that might get in surrounding the neurons that might damage them. So bacteria, viruses, it will take care of, dispose of any dead neurons that might be in our brain. Even though we're born with all the neurons we're ever going to have, we die with a lot fewer neurons than we were born with because they die and don't ever get replaced. And so we want to make sure of course, we don't do anything to hasten the death of our neurons, but if the neurons were to die, then the microglia would take care of that. So these are phagocytes or macrophages that embryonically left the bone marrow and went into our brains and then stay in our brains for the rest of their existence. Okay, so they hang out in our brain tissue. All right, then we'll go to these cells. These are the ependymal cells, so they're the next ones on our list. So ependymal cells are going to line fluid-filled cavities. We haven't learned that there are cavities within our brain and spinal cord, but there are. And so in those cavities, we need to move around the fluid that surrounds the neurons called cerebrospinal fluid, or CSF. And so in order to circulate that CSF, these ependymal cells have cilia on the ends of them, and so they will sweep and create a current that will help circulate the cerebrospinal fluid. So that's their job. They line fluid-filled cavities to circulate cerebrospinal fluid. All right, now yesterday I mentioned about saltatory conduction and the fact that if you wrap a protective lipid material called myelin around the axons that it protects them, and it also speeds up the rate of transmission. So we have two kinds of cells in our nervous system that will produce myelin. The ones that are found in the central nervous system, right here, are called oligodendrocytes. So an oligodendrocyte is a cell that will wrap itself around multiple axons. So here you can see I've taken one cell and I've wrapped it around numerous times. If you look at the picture on 252 of oligodendrocytes, they kind of look like, I wish I had that picture. I don't have that picture in my collection here. I'll have to find where it is. They look a lot like the Enterprise from Star Trek. And so you can see one oligodendrocyte is wrapping around multiple axons, and they just kind of stack up on one another and create the myelin sheath that we find within the central nervous system. Okay, so remember, oligodendrocytes, central nervous system, and then in the peripheral nervous system, so out in the nerves of our body outside of the brain and spinal cord, then we have Schwann cells that are going to produce the myelin. So if we look at this one axon, here we can see multiple Schwann cells that are surrounding the axon. So it's just opposite to an oligodendrocyte as far as number, like cell ratio. So you get one oligodendrocyte to several neurons, but in the PNS, you get one neuron with many Schwann cells on it. Notice that the Schwann cells don't touch. So we have those nodes of Ranvier. We'll talk more about those today. And the Schwann cells are going to wrap their entire self around the axon to create the myelin sheath. And I was going to say something else, and I just don't remember what it is. It'll come to me. So remember then, Schwann cells in the PNS. We are not going to worry about satellite cells. They're the ones that are going to surround the cell bodies of the neurons that we find out in the PNS, but we're not going to worry about that. So Schwann cells, PNS, oligodendrocytes, let's go back to that picture, oligodendrocytes, CNS. So remember that relationship. Okay, so those are the neuroglial cells. So now we're going to go to the structure of a neuron. So there are three parts to a neuron. We have this receptive end that are the dendrites. I don't know why it cut off. It's up there, but anyway. So the dendrites are the receptive regions. This is the main place where the incoming stimulus is going to reach the neuron. Then we have the cell body, which is the center part. And so it says biosynthetic and receptive region. Don't worry about the biosynthetic part, but receptive means it can also pick up signals. So when we were looking at synapses in lecture yesterday, that we saw, oh yeah, an axon can synapse with the dendrite, it can synapse with the cell body, and it can synapse right at the beginning of the axon. Okay, so that's the cell body. It's also known as the soma, but I'm never going to call it that. So just cell body is fine. So dendrite, cell body, and one axon. So notice here I have numerous dendrites, and so that's going to increase the likelihood that a neuron is going to pick up a signal from somewhere else, and then it'll be sent through so that depolarization will happen through the cell body, and then it'll reach the single axon. And there is only one axon on every neuron. There are never any more. You may have some branches way down here at the bottom or some collateral branches, but you're always going to have one axon that leaves the cell body, and the signal will always and forever be sent dendrite, cell body, axon. It will never ever go back the other way. Okay, so dendrite, cell body, axon. So in the axon, make sure you add this word to your list. Okay, axon hillock is this little spot right here. And the purpose of the axon hillock, remember, is where we start to get the voltage gated sodium channels begin right here. So this is the first place that we get our voltage gated sodium channels, at the axon hillock. Okay, so here's the one long axon, and then the very beginning of it is called the axon hillock. All right, the myelin is just the lipid material that's surrounding the axon, and only axons are myelinated. Dendrites and cell bodies are never ever myelinated. And what else do I want to say? It's made out of the cell membrane, so it's primarily lipid material. And so the Schwann cells here will form the myelin. I think that's about it I want to say about that. Okay, so because the Schwann cells never touch one another, then we have these gaps of bare axon called nodes of Ranvier, or myelin sheath gaps. And so the propagation, so the action potential, because the sodium can't flow in at any other spot along the axon other than at the axon or at the nodes of Ranvier. So that's where the depolarization will happen. And so it'll depolarize, and it'll depolarize at each one of these gaps, and that will speed up transmission down the axon, and we call that saltatory conduction. And then the synapse, of course, is just at the end between the presynaptic neuron and the postsynaptic neuron. All right, now neurons come in, okay, so neurons come in different types, different flavors based on their, I don't want that. Let me see. Based on their structure and their function. So we're gonna go there now. And let's see, in your notes, oh, there it is. Go to page 255, and we have classified structures, and then 257 shows us the classified according to their functions. Okay, so let me find my, oh, here we go. All right, so which do we want to do first? We want to do structural ones. So your pictures are going to look kind of like these. Yep, you have these. Let me just go back a picture. Okay, yeah, because you have these pictures on the top, well, in the very middle of the page on 255. Okay, so we're going to look at types of neurons based on their structure. So, and then I'm going to go to the whiteboard, and I'm going to use my lame mouse, and I'm going to draw you some as well, but I'm going to introduce them, and then I'm going to go to the whiteboard. Okay, so we got three kinds. If we look up here, we have multipolar neurons, we have bipolar neurons, and we have unipolar neurons. Okay, the pseudo-unipolar, don't worry about that, just unipolar is good. Okay, so what are some key things about these neurons? Okay, so we have many processes, okay, all are dendrites except for a single axon. So if we look at here, we can see many, many dendrites, but only one axon. So this is a multipolar neuron. Okay, a bipolar neuron, we have two processes extend from the cell body. One is a dendrite and the other is an axon. So here we have just a single dendrite, boom, with its little branches. So the signal would be going this direction. Okay, and then we get to the cell body, and then we have an axon, and then all its little branches. But again, our signal is going to be traveling that direction. So one single dendrite, one single axon. Okay, then in a unipolar cell, what we have is one single axon. So this is kind of weird. Where are my dendrites? We don't really refer to them as dendrites, but these are the little branches that are at the beginning because they're receptive endings, so they're at the start. Okay, and then so we have a cell body and peripheral processes that make the whole axon. So we have peripheral, cell body, central process, but you don't need to remember these names, so don't remember those names. I don't care. All right, but the one thing that we do want to notice here on the axon is that the cell body doesn't have dendrites on it, and it does extend out from the axon. So the cell body is actually away from the axon and not in line with the axon, like we have here and we have here. So the cell body is off to the side. And notice that in this one, the unipolar or the multipolar neuron, that we have myelin on the axon. We have no myelin in the bipolar neurons, and then we have myelin again, whoops, in the axon of the unipolar neuron. Okay, so those are the structural types: unipolar, bipolar, and multipolar. Okay, now we're going to look at functional kinds. So we're going to go over to either 257. You can, yeah, 257 is the one that I want you to look at. So I am going to go to find the picture that I want. Okay, I'm going to do this one first, and then I'm going to go to the picture similar to what you have in a reflex arc. Okay, so remember that we had 3 jobs or 3 things that a neuron did, or 3 parts to our nervous system. So we had sensory input, which was blue. Okay, so we'll draw a little thing out here and some little endings. Okay, so that would be my sensory input, like that. Then I have, do that one. Then I have my integration. I'm going to draw this and then down like that. Okay, so this is where integration happens. And then my last one is motor output. Okay, now this is my motor output. All right, so now when we talk about functional, we're going to also say, well, what cell shape does the job? So my 3 jobs are to be a sensory neuron. Let me get a different color. Okay, so I'm a sensory neuron. So here we go. Sensory neurons conduct impulses along afferent pathways to the CNS. So here is my sensory neuron. You can see the signal is going this direction from out in your body to the central nervous system. Now, what shape of neuron are we going to use? Well, if we look at this one, it has this long thing, and then we have this cell body that's out to the side right here. So that is a unipolar neuron. Okay, so almost all sensory neurons, that's its function as a sensory neuron, carry signals to the CNS. And most sensory neurons are unipolar, have that shape. And so that's why I drew it like this over here. Okay, then bipolar neurons are also sensory neurons, okay, but they're not very common. So I'm going to put not common. So sensory neurons can be both bipolar and unipolar, but for all intents and purposes in here, a sensory neuron is always going to be unipolar. Okay, so now we've only dealt with one functional type of neuron, the sensory neuron. So now we need to take a look at this one, the one that's in integration here in the brain and spinal cord, and that's called an interneuron, because their job is to conduct impulses within the CNS, which means they're integrating sensory input with motor output. Okay, so this green little jobby is an interneuron, and it'll only be found in the central nervous system. We don't have interneurons anywhere else but in the central nervous system, and they're only going to be in this brown stuff, which is really not brown. Well, it actually really is brown, but its name isn't brown. We don't call it brown matter, we call it gray matter. Okay, so interneurons are found entirely within the gray matter. They are not myelinated, and structurally they are multipolar neurons. So they're going to have lots of dendrites up here at the top in the cell body and one long axon, and they're going to synapse with somebody else. So they're going to have this shape with the cell body and then dendrites. But they're just going to be little tiny baby ones. Okay, and then motor neurons, these guys right here, that are going to carry the signal from the CNS to an effector, so either a muscle or gland, are motor neurons. And then structurally, they are also multipolar. So this red guy right here looks just like this. Okay, so this red one is a multipolar neuron too. All right, so going back to our yellow packet then, that our functional neurons, sensory neurons, carry impulses from out in the body to the CNS, carry signals toward the CNS, and they're going to be unipolar or sometimes bipolar, but mostly unipolar. Interneurons are going to be found in the CNS between sensory input and motor output, and they're going to be structurally, they're going to be multipolar neurons. And then motor neurons are going to carry signals from the CNS out to the body, and they're going to be multipolar neurons as well, structurally. Okay, so that's that. All right, now I need you to go to, and then, oh, so let me just go to the picture that's kind of like your 257, is this one right here. So they've drawn them just exactly the same. So we'll just label which kind these are. Oops. Okay, so this is a, well, sensory neuron. Okay, so it's sensory, so it's going to be a unipolar. Okay, this is an interneuron right here. Whoa, that was big. And so that's going to be a multipolar. Okay, and then this is the motor neuron, and so it's going to be multipolar too. All right, so in this picture you have both the functional classification and structural, except we just don't have a bipolar neuron. So bipolar neuron looks like this. Color that all in. And nucleus in the middle. Okay. And I don't think I need to go to the whiteboard. I think we've looked at enough pictures there. Okay. Now, the next thing on your list is the structure of a nerve, because we're going to be looking at nerves today too. And so go to page 258. And I'm going to have to go to a different chapter for that picture for myself. Where's the nerve? Oh, right here. Oh, actually, I like this picture. Let's go back to this picture. All right, you don't have this one because your photomicrograph is just a microscope. It's just a regular photomicrograph that you have on 258. I have scanning electron micrograph, and so it's actually kind of neater, cooler looking. But let's do this picture, and then we're going to come back to this picture. So, okay, so we have the same kind of organization in a nerve that we have in a muscle as far as connective tissues are concerned. So let's do whole muscle, whole nerve right now. So remember, around an entire muscle was called epimysium. So now around an entire nerve is going to be called epineurium. Yes. Okay, so here's the epineurium that's going to go around the entire nerve. Within the nerve, we're going to have nerve fibers. So we're going to have axons here. And so here, and they're just axons. There are no cell bodies and there are no dendrites in a nerve. They're just axons. So we're going to bundle those axons together, just like my muscle fibers. We're going to bundle them together into fascicles. And the connective tissue that's going to wrap a fascicle in a nerve is called perineurium. And then each neuron itself, each axon, is going to be surrounded by connective tissue called endoneurium. And then endoneurium is going to go around the myelin sheath, and then the myelin sheath is going to go around the axon. So these guys are pretty protected which you want them to be, because they're out there in the PNS. They're out there for you to hit your elbow and bash your ulnar nerve and hit your funny bone and make it feel weird and everything. So we have some decent protection around our axons or in our nerves. So let's go to the picture before and let's find all of those connective tissue coverings. So I'm going to make my pen really, let's do purple because that's going to show up pretty good. So we're going to do purple, and then I need to do it super, super thin, at least that we can still see it. Okay, so in the center of a nerve, we got this blood vessel right here. Here's all the red blood cells inside of there. All right, so let's go easy first. Let's start from the outside. So here is epineurium all around the outside. Okay, so all of that. Oh, I'm not drawing very gently. Okay, all of that is epineurium. Now we want to bundle the axons together into groups called fascicles. So here is going to be a fascicle right there. And then I have a fascicle that's going to feed into that perineurium. And then I have a fascicle here. And there's some perineurium right there. Okay, so there are, and I don't, this is hard. I can't really tell where the fascicle is, where the perineurium is in this section. All right, then I have endoneurium. And so in my little Cheerio looking things, the endoneurium is going to be right around the outside, but okay, here you can get it on this one. All right, but see how there's still stuff inside the perineurium or inside the endoneurium? And so the thing that looks primarily like a Cheerio is the myelin sheath. It's going to go right around the opening, and that hole right there in the center is going to be the axon itself. Okay, so I like this picture because we're able to see all those different layers and different connective tissues a little bit better than what is a little easier to see than what you see on page 258. Okay, so we're gonna, that's it for this list. I'm gonna do the neurotransmitters tomorrow, and I think we're good on EPSPs and IPSPs. Just remember, oh, let me go to my whiteboard and we'll just make sure we have some little definitions here. Okay, so okay, so an EPSP, oops, PSP, okay, is a excitatory postsynaptic potential. So these are graded, which means they come in various sizes, graded local potentials that increase the likelihood of an graded local depolarizing, I left out a word, depolarizing potentials that increase the likelihood of an action potential being produced at the axon hillock only present on dendrites and cell bodies with chemically gated channels. Oops. Okay, so that's an EPSP, and then an IPSP. IPSP is an inhibitory postsynaptic potential, which is again a graded local, oh my heavens, you guys, local hyperpolarization that decreases the likelihood of an action potential being produced. Okay, and then again, only on the dendrites and cell bodies with not chemically gated channels. This, well, yeah. Oh, I know what I want to put. Let's see. Neurotransmitters that are hyperpolarizing will open potassium and chloride channels. Okay, so here we're opening sodium channels. Okay, and then in the other ones, then we're opening potassium and chloride channels to hyperpolarize them. Okay, and then synaptic transmission, that's just the events that happen at the synapse, and then the neurotransmitters we'll do tomorrow. So I'm not gonna quiz you on neurotransmitters. So you can cross that off your list if you want. Okay, so that's all of that side. So now we're gonna go over to week 14 in the Thursday lab, so we just did that from week 12 in the Monday lab. And then, yeah, and don't worry about, on the Monday lab, I have nerve impulse events. Yeah, don't worry about that either. So now we're gonna go over to structure of the spinal cord, reflexes and nerves in week 14. And for Thursday lab, we're on week 13. But structure of the spinal cord, reflexes and nerves. And I'm just gonna cover this now, but I'll give you another week to do the homework. I'm not gonna make you do 6 homework assignments in a week. So we'll push 19, 20, and 21 out a little bit farther, but I do need you to be working on 15, 16, and PEX3 for next week, okay? All right, so now we wanna go to the structure of the spinal cord. So I'm gonna get rid of that and share the spinal cord. So let me get rid of that and go back to 12. And okay, all right, so I'm gonna go, so now we want to be in exercise 19. Okay, so on 302 and 303 and 304 are the pages that we're going to be taking a look at right now for structure of the spinal cord. So let me find my spinal cord pictures. Okay, there it is. Couldn't find that for a minute. Okay, so here is the 2 pictures. So the one on 302 and the one on 304. Okay, so let's take a look at those and then okay. And then we'll go back to some more about the spinal cord. So let me get that sharing for you. Okay, here we go. All right, so let's look at this view right here. So this is a cross section through the spinal cord and vertebra. And just to orient you, we need to make sure we know which is front and which is back. So this is anterior right here. So this is the front side. This is posterior back here because this is the spinous processes that are on the back. This is right and this is left. Not that that matters too much. I don't really care about that, but you need to know which is the front and which is the back. Really important. Okay, so let's start out with gray matter. So gray matter is everything that's unmyelinated. So if we look in here, this brown stuff right here is gray matter. And gray matter is made out of the interneurons. So interneurons okay, so that's gonna be in there. And then the cell bodies and dendrites, because dendrites always come before cell bodies. And then cell bodies of, don't know what I was trying to do there, motor neurons. Okay. Oh, it hurts my brain. All right, so let me just show you how that's going to work. So I'll have some input. So this is where I'm going to have sensory input and then integration and motor output. So sensory input is going to come into here, into this posterior horn, and then I'm going to have an interneuron here, and then it can either synapse with a neuron in this lateral horn or in this ventral horn right here. And then that's where the interneuron, I mean the motor neuron is going to start, and then it's going to come out here. And so sensory came in the back, and then motor comes out the front. Okay, so that's the gray matter is going to be the interneurons in the spinal cord and the cell bodies and dendrites of the motor neurons. Okay, next week after we take our next exam, when we start in on the brain, we'll be talking about how did our nervous system form, and it forms from a tube, from a hollow tube. And so the remnant of that hollow tube is this little circle in here. I don't know why they didn't label it here, called the central canal. Central. Okay, now remember we said we have some ependymal cells that are going to move cerebrospinal fluid. Well, there's cerebrospinal fluid that runs through the center of the spinal cord, and so the central canal is lined with ependymal cells in order to circulate the CSF down through the cerebral, or I mean down through the spinal cord. Okay, so central canal lined with ependymal cells. Okay, now we need, because we have our sensory input, integration, and motor output and everything, we need a way for sensory input to come in and integration to happen and motor output to go out. So we have two roots that are going to come, and that's probably in the different picture, but I'm still going to use this one anyway. Okay, so I have these roots. I have a root that goes in this way. Okay, so that's going to be sensory input, because sensory goes in here. And because this root is on the back, it's on the posterior side, we call this a dorsal root. So dorsal root, and we'll talk about what a ganglion here is just a minute. Okay, so dorsal root is for sensory input. Okay, so dorsal root is sensory. All sensory input comes in through the dorsal root of a spinal nerve. Then I'm going to integrate, and so my interneurons are going to be in my gray matter of my spinal cord where my integration happens. And then my axons of my motor neurons are going to come out through the ventral root. So they're going to come out here on the front side. So ventral and anterior are the same. And that will always be motor output. So the ventral root is always motor. Oops. Okay, now remember, so dorsal root is sensory neurons and ventral root is motor neurons. Now remember how in a sensory neuron or a unipolar neuron that you had this cell body that comes out like that? Okay, so what we're going to do is cluster all of the cell bodies together of every sensory neuron in this dorsal root right here. So in this dorsal root right here, I'm going to put all the cell bodies together. So I'm going to have an axon that comes in, and I'm going to put a cell body there, and then I'm going to send the axon here. And then I'm going to bring in another neuron and put another cell body there and another axon. And I'm going to keep doing that, but I'm going to put every cell body in the same place right here. And when I have cell bodies that are outside of the gray matter of the brain and spinal cord, then I call it a ganglion. So let me get rid of all of this. Okay, so this fat thing right here, and notice, oops, notice that it's only in the around the dorsal root. Okay, so here's the dorsal root, here's dorsal root here, and it's only fat in the dorsal root. So there's only a ganglion in the dorsal root, and it's going to be the cell bodies of the sensory neurons coming into the spinal cord. Okay. All right. Now, that's all of the spinal cord that you need to know. So let's take a look at this other picture. This shows a little bit better. So let's draw some things in here. Let's put them in their respective colors. Blue. Okay, so let's do our sensory input. So our sensory input's going to come in through one half, and we're going to put a cell body there, and then we're going to put it there. Then we're going to bring another one in, and we're going to stack it up here, and then send it on in there, and so on. Okay, then we're going to have interneurons that are going to be in here, that are going to synapse over here, and more interneurons. Okay, and then they are going to synapse. So we had the sensory neurons coming in the dorsal root, and then now I'm going to have my motor neurons coming out my ventral root. And then I'm going to have a neuron here. He's going to go out through there. Another neuron here, and he's going to go out through there, and so on. Okay, so dorsal root, sensory input, ventral root, motor output. And then here's the central canal, boom, and the gray matter, just gray matter all together. You don't need to know the measures or the 3 kinds of horns, dorsal, ventral, lateral horn. You don't need to know that. Okay, so that's all of that part. All right, now we want to take a look at the other nerves that we find with the associated with the peripheral nervous system. So let's do this picture right here. All right, so this is the picture that you see on page 303. So this shows the spinal cord, and if your micrographs are a little bit different, but that's okay. So we've got some regions of spinal nerves. Oh, first of all, we need to know that there are 31 pairs. Okay, so there's 31 pair of spinal nerves running down here. I'll add them all up, there'll be 31 pairs. And the 31 pairs are have both a sensory and a motor part. So they're mixed nerves, both sensory and motor in the same nerve. They're just going to be like a divided highway. Okay, so all the sensory neurons will be together on one side, all the motor neurons will be together on another side. All right, so we have these 3 regions. We have a cervical region, and notice that it gets a little fat in here. Then we have a thoracic region, and we have a lumbar region and a sacral region. Now, one of the things that we notice when we get right into the beginning of the lumbar region is that we cease to have spinal cord. So see, we have spinal cord in the cervical region, we have spinal cord in the thoracic, but our spinal cord ends right here at what's known as the conus medullaris. So this is the end of a solid spinal cord right there, conus medullaris. Now all the nerves are going to fan out off of here without a solid spinal cord, and we're going to call it the cauda equina. I know I don't have that on the list, but you're going to eventually be responsible for it, so I'm telling it to you now. Okay, so spinal cord ends at the start at L1 at the beginning of the lumbar region, and then you just have nerves that come out that look like a horse's tail, so we call it cauda equina. But these nerves up here when they come out, and then down here in the lumbar region, they come out kind of interestingly, that I'm going to show you. And that's the rest of the nerves that you have on your list. And I'm going to have you add a couple of terms. Thursday has one of the terms. Monday doesn't have either one of them. So I'll have you put them on in just a minute. Okay, so we're going to go to this cervical region, actually more arm region, brachial region, to a collection of nerves that's not on this picture. I'm going to go to a different chapter. Hang on. Called, hang on. There we go. Okay. All right, so let's go to this picture. I think you guys have this. Maybe not. Oh yeah, you do. Go to page 307. And now we're going to look at what are known as plexuses. So a plexus is where nerves come out and interconnect with one another. And so as you can see up here in the cervical and brachial regions of your body, we have these definite interconnecting of nerves. So we have cervical plexus, brachial plexus. Look at how these nerves are all, they're all kind of interconnected with one another right there. Then we don't see that happening in any of the thoracic nerves, but then we see it picking up again and doing it in the lumbar and sacral region of your spinal cord. So a plexus is just a section of your spinal cord where the nerves that come out, the ventral roots that come out, all interconnect with one another. They just don't do it in the thoracic region. So we need to look at these plexuses. Okay, we don't want to look at that. Okay, so cervical plexus. So we're in the cervical plexus right now. Ooh. Okay, and there's a nerve. So I want you to add cervical plexus. Actually, I guess there's 4 things I want you to add. All right, so look at all this crazy interconnecting weirdness thing. It's like, look at 1 comes out and it loops around, it goes back into 2. How weird is that? I don't know. But there's 1 nerve that I want you to know right here that comes out of the cervical plexus, and that's this one down here called the phrenic nerve. And the phrenic nerve goes to your diaphragm. And this is the one that tells your diaphragm to contract in order to breathe. So now let's say that I had a neck injury. I was in a car accident, or I was riding a horse and I fell off the horse or something. And I had an injury between maybe C1, C2, or C2, C3. Okay, if we look at how, if I damaged and these nerves weren't working anymore, how everything downstream from that would be affected. Okay, so if I break my neck up high, I'm going to be on a ventilator for the rest of my life. I am not going to ever be able to breathe on my own. Okay, so damage to my spinal cord at C1, C2, C3 is, you're probably going to end up, you'll definitely be quadriplegic, but you won't breathe on your own. You will always have like a trach tube and ventilating for you. Okay. If we break down here like C6, C7, that's not going to be affected. Okay, because those nerves do not contribute to the diaphragm. Not this one. Erase that one. But if I damage my spinal cord below the level of C5, I don't have to be on a ventilator. I will probably be a quadriplegic. So both my arms and legs will be paralyzed, but I will be able to breathe on my own under most cases. Okay. All right, so that's cervical region. Now we want to go to the brachial region, to your arm region, to what's known as the brachial plexus. All right, so make sure you have brachial written on your paper. If you don't, Thursday does, but okay, we can write. Ooh, no, I need a different color. I need purple. Okay, so this is the brachial plexus. Now, we have nerves that are coming out of your neck. C4, 5, 6, 7, 8, and T1. So more nerves coming out of your neck and one from your thoracic region, and they interconnect right here and form 4 nerves. Okay, so the first nerve that you need to know, and so you'll need to add this, is the musculocutaneous nerve. And this one's gonna tell your upper arm to contract. So it's gonna control your upper arm muscles, your biceps brachii and your brachialis and your brachioradialis. Okay, so musculocutaneous. Now the radius, the radial nerve, okay, that goes along the back. That's why it's green, okay, because it's posterior. So it's going to run along the back of your arm, and it's going to do all of your extensor muscles down into your wrist. Extensor, okay. So remember, we learned the extensor, carpi, radialis, longus, and brevis, and all the extensor muscles on your back of your arm are going to be innervated by the radial nerve. Now the median nerve, okay, this one is going to go to both of the radial and ulnar are going to go to your flexors. Okay, but the median nerve is going to go to fingers 1, 2, and 3 and part of 4. This part of 4. Okay, so your thumb, your index, your middle finger, and the inside portion of your ring finger are going to be innervated by the median nerve, and this is the nerve that gets pinched off in, oh, you guys, I just lost it. I've been thinking about it this whole time. Carpal tunnel syndrome. So the median nerve runs right in the middle of your carpal bones on the flexor side of your wrist, and so if you bend your wrist, like you extend your wrist too much, or hyperextend your wrist, actually, it's going to pinch that nerve in there, and then you get carpal tunnel. Okay, so carpal tunnel affects the median nerve, and then the innermost nerve is the ulnar, and that's going to go to part of 4. Sorry about that. Part of 4, so part of your ring finger, and then also your pinky finger. And remember, that's the one that's going to run along the inside of your arm on the ulnar side when you're in anatomical position. And when you hit your funny bone, that's the nerve that you're hitting, is the ulnar nerve. All right, then we have the lumbar plexus or lumbosacral plexus, and that's going to be way down here in your hip. Okay, so one more. Okay, here's where we're going to go. All right, so sacral plexus. We have one big giant nerve right here in our sacral plexus, and that is the sciatic. And so the sciatic is going to run down the back of your back of your leg, and cause your hamstrings and down in towards your calf muscles in order for them to contract. So some of you may have a parent or a grandparent that has sciatica. So they have like a pain in here. I had sciatica with my first pregnancy, my son, because this comes out of by the greater sciatic notch on the os coxa. And so he would push in here because this is inside my pelvic cavity. And so he was such a big kid that he would compress that nerve, and then my leg would just give out and I'd fall down. And so that wasn't very fun because I was big and fat and I couldn't get back up again. And so his dad would have to come and help me up off the ground because I was so big. If we look at how that sciatic nerve runs down the back of your leg, oh, yeah. There it is. Boom. And then it gets to your knee and it goes to the common fibular and tibial right there. Okay, so that is the sciatic nerve, the largest nerve that you have in your body. All right, just a few more things and easy things and then a hard thing and then we're done. Yay, thanks, Ms. G. All right, so now we are going to go to the reflex arc. So you're going to go to exercise 20, not 20, exercise 21. Yeah, exercise 21. And we're going to look at the picture, figure 21.1 on page 330. So I'm going to find that. There it is. Okay, so this is a reflex arc. So you need to know the 5 parts of a reflex arc, and we really have already gone over them so much that it shouldn't be new to you, because the reflex arc really demonstrates how your what your nervous system does. Because remember, we had 3 parts: sensory input, integration, motor output. So we only need to add a couple more things to those 3 things, and then we have a reflex arc. So the 5 parts of the reflex arc in this order. Okay, so always remember, number 1 in a reflex arc is the receptor. So that's going to be, it could be an actual structure to a receptor, or it could just be dendritic endings. It depends. Okay, so, but here is the receptor. Then part 2 of a reflex arc is the sensory neuron. So here is that unipolar neuron coming into the spinal cord. Then 3 is the integration center. Now, what do I have in an integration center? Well, it's going to be at the level of the spinal cord. Okay, so we have that. And there will be an interneuron. Now, there doesn't always have to be an interneuron. Sometimes there's not an interneuron. Sometimes the sensory neuron will synapse immediately with the motor neuron, but sometimes, sometimes there's an interneuron there. More times than not, there's an interneuron. Okay, so at the integration center will be an interneuron. Oops. Okay, there we go, make that prettier. Is an interneuron, and then where is the interneuron found? In the spinal cord. Okay, then we need to send the signal back out, so we have a motor output, so we have a motor neuron, so that'll be the 4th part of a reflex arc, and then the last part of the reflex arc will be the muscle or the gland that the motor neuron is stimulating. Okay, so 5 parts of a reflex arc: receptor, sensory neuron, integration center, motor neuron, and the effector. Okay, so there's those parts. And lastly, look at page 331 just really quickly. If you see in the picture on the left, the reflex on the left, you can see there's no interneuron. But the reflex on the right, there happens to be 2 interneurons there. So there just shows how you can have, and we'll talk more about this for the next test in the lecture part of the class. All I want you to know right now are the 5 parts of a reflex arc. Okay, last but not least are the cranial nerves. I have to think what they're called. So let's go to this picture, and this is going to be back in 17. In exercise 17 in parts of the brain on page, oh heavens, oh my goodness, where is it? Oh, there it is, 280. So on page 280, this is the underneath side of the brain. This view is the underneath side of the brain. And this view is, what was I going to say? Oh, and so these are the nerves that come out of the brain. So all the other nerves that we looked at come out of the spinal cord, but these come out of the brain. So we call them cranial nerves. Now, let's see. I'm trying to decide. Normally I make you do a chart for this, but this chart, we're going to add to this chart. Okay. So let's talk about this as we go. And then I'm going to post, I think I'll just go ahead and post a document, a little handout thing that I normally have you fill out as we go through this stuff. But you know what? We can wait and save that for the lecture stuff. So all we're going to do right now is just identify the cranial nerves. That'll be good enough. And then we'll talk more about their functions when we get into lecture. Yeah, I think we'll do it that way. Okay, so I'm not going to post a document. All I want you to do for next week is to be able to look at this picture and tell which things are which. There's a bunch of yellow sticky outy things. How can I tell which is which? Well, I'm going to teach you that right now. So here we go. So get used to this picture. What you may want to do with your lab manual is just cover over the list. So where all the words are, and practice just going down through the list, touching every single one of the nerves until you get them down, because this can be really tricky. But the first thing that you need to know is that they come in a particular order. So we put Roman numerals next to the side of them that indicate where they come out of the brain. I mean, at what, who comes out first, then who comes out second, and so on. So we're going to use that to help us figure out what each one of these nerves are when it gets down here by the medulla and it gets really tricky. All right, so the first cranial nerve is known as the olfactory nerve. So I'm just going to draw some things with this and make it a little skinnier. Okay, so olfactory nerve right here. So this is one. That's the bulb, but it's this whole thing right here. So I'll write next to them as we go so that you can, I don't know if it's going to help or not, but anyway. So I do want you to know that the olfactory nerve is for smell. That's what olfaction is. Okay, so smell. The next nerve is the optic nerve, or nerve number 2. So this is 2 right here. This is 2 right here. Now what happened, and so then they would go on out to the eyeballs, out this way. So then you'd have eyeballs on the end of your optic nerves. When they come in, okay, because they're going to bring sensory input in, that they're going to cross. And so some of the signals are going to go to here, and then some of the signals here, and then input from your eye is going to go over here and then come down here. So you can see, oh yeah, look at that. I got some input coming here and input coming there. And so they cross. And when we do vision, we'll talk about, well, why do they need to cross like that? Okay, so that's the optic nerve. So olfactory, optic. The next one is oculomotor. So this is oculomotor number 3, right down here at the base of this thing right here is called the pons. Just put a little P right there. And this is for eye movement. Eye movement. So it's there's actually 3 cranial nerves that are responsible for moving your eyeball around. So it does more things we'll talk about later, but just for now, you need to know eye movement. Okay, trochlear nerve is number 4, and it also is eye movement. Okay, so oculomotor and trochlear are for moving around your eyeballs. And then we have 5. Okay, this is the trigeminal, cranial nerve 5. Now, they didn't put this in the picture, but on our brains that we have, and I'm going to put it over this side because I'm not really drawing. The cranial nerve is going to come off like this, and it's going to have 3 branches to it. Oops. Okay, so the cranial nerve is both sensory and motor. So we're going to have one part that goes to your eye, like if you get poked in the eye, and then your upper jaw and your lower jaw. So really all sensation on your face comes through the trigeminal nerve. Okay, so everywhere on your face, and poking in the eyeball and everything, all that's going to come through the trigeminal nerve. And then it's also, because it's associated with the mandible, it's going to be for the muscles of mastication. So we use it for the masseter and the the temporal. So it's also for chewing. Okay, so our sense of feeling in our face, and then our chewing muscles is trigeminal nerve number 5. And then our last one of the first half is the abducens nerve, cranial nerve number 6, and this is also for eye movement. So these are the three the three nerves that go to the muscles of the eye to move the eyeball around. So ocular motor, trochlear, and abducens. All right, now we'll go to the other side of the brain and over here where they're labeled, and this is where it starts to get tricky. So I'm going to put 6 right here, okay, because this is still an abducens. And oh, by the way, all of our cranial nerves are paired. So we have 12 pairs of cranial nerves. Okay, so the next one is the facial nerve, cranial nerve number 7. It kind of looks like it's a pair of nerves, okay? And this one is for the muscles of your face. So remember when we were looking at all those pictures, facial muscles, there we go. And you got Bell's palsy. This is the nerve that's affected if you get Bell's palsy. So this is what controls all your facial muscles. So I'm definitely using it right now because I'm moving my orbicularis oris and my buccinator and my I don't know, whatever else I'm using to speak to you. So my facial nerve, and it's also for taste. Oops. So the front of your tongue, the sensations in your tongue, you bite your tongue, it's hot, okay, all of those things are going to go through your facial nerve. All right, the next nerve is right so close next to it, right here, that sometimes it gets a little tough to see. So I'm going to put 8 over here as well, because that's a little bit better that you can see. So cranial nerve number 8, vestibulocochlear nerve, is hearing and balance. So you're using your vestibulocochlear right now. Okay, so hearing and balance. And then cranial nerve number 9 is glossopharyngeal. This is number 9. Okay, this is swallowing. So your nerves in your throat for swallowing, and part of your tongue, and also taste in the back of your tongue, in the back of your mouth. When bitter, yucky flavors go back there, it's glossopharyngeal that's taking in that information. Okay, vagus nerve number 10 goes to all your internal organs, and this is primarily your parasympathetic. So your feed or breed response. We'll get more into that later. Okay, but we're just going to go all of your muscles. I mean, all of your internal organs, not any muscles, all your internal organs. Now, let's say that you're having a heart attack. It's your vagus nerve that's going to be bringing that information into your spinal cord. But because the nerves from your arm, from your left arm and down the left side of your body, come in at the same place in your spinal cord as your heart sensation, the pain from your heart, your brain thinks it's coming from your arm. So we call that referred pain, because your brain is so not used to getting signals from your heart, and it's been getting signals all your whole life from the left side of your body. So it thinks that you're getting it from the left side, and so the pain is referred to that area. And so that's why we feel heart attacks on the left side of our body. The accessory nerve is number 11, and this is going to go to shoulder muscles. So shrugging your shoulder muscles, shrug like your trapezius. Okay, so your shrug muscles. And then last but not least is cranial nerve number 12, and hypoglossal goes to stick out your tongue. Sticky, sorry, stick out. Oh my gosh, I'm tired of writing like this. Stick out your tongue. You need to get a stylus or something that would write. Woo. Okay. Fabulous. So what's gonna be, look at that picture, that's gross. Okay, so what's gonna be on your quiz next week? So, well, your quiz that's due next week. So it's gonna be the neuroglial cells, parts of a neuron, types of neurons, structure of a nerve. That's so easy. Those are just super easy. And then structure of the spinal cord, parts of a reflex arc, super, super easy. And then the only hard part is making sure you can identify the 12 cranial nerves. Functions won't be on there. All it is there'll be an arrow to it, and I'll just say, tell me which cranial nerve this is. Okay. All right. So that's all I have for you today. Does anybody have any questions before we go about what's going to be? So it's just pictures. It's all the same pictures that you guys have in your lab manual. Okay. Well, if we don't have any questions, then I'm going to just go ahead and stop my recording, and I'll get this posted. And let's see. So I'm not going to post the cranial nerve thing. We'll do that in lecture. So I'll just post this, and then the notice about the blood drive on Thursday. All right. Okay, you guys have a great week, and well, I'll see you tomorrow. We'll see you tomorrow. Bye.